And I'm going to focus on the use of CTCs and CTC analysis to study cancer metastasis. My disclosures, the work that I'll describe today was in part sponsored by Janssen Diagnostics, which has given us funds to build the CTC chip that I'll describe today. So this is the challenge of circulating tumor cells, as well known. These cells are shed by both primary cells as well as metastatic deposits. They enter the bloodstream, and then they can travel. And most of them die in the circulation, so they're useful as markers of cancer, but they themselves may not have the potential to give rise to metastases. Some of them will. Some of them will extravasate, give rise to metastases, and cause most of the lethality of cancer. The challenge is shown in this slide, which is that a tube of blood will have 10 mils of blood, will have 50 billion red blood cells, 50 million white blood cells, and anything from 0 to 100 CTCs. So if you look at this as an engineering challenge, it's really one that has benefited tremendously from microfluidics and the application of novel cutting-edge bioengineering to try to find these rare cells. And these are rare cells in transit. So if you think about most of what we've learned in metastasis, it's by looking at the primary tumor, looking at the metastasis, and seeing how they're different. The ability to study these things in transit is biologically very, very interesting. You've heard a talk yesterday from David Miyamoto in the group who's used the same chip, so I'll go through this relatively quickly. But this is the idea of, this is our third generation chip, which we call the I-chip for inertial focusing. And what you can see with this chip, it was developed by Mehmet Toner, a long-term collaborator and bioengineer. And the idea is that the chip microfluidics is so powerful that we can actually get rid of the white blood cells and the red blood cells and leave behind the untagged tumor cells. And that's a big change compared to having to fish out the tumor cells based on assumptions about what they do or don't express. And to show you through the mechanics relatively quickly, you start with a tube of fresh blood, and you add antibodies against white blood cells. And these antibodies are magnetically conjugated. Now, everybody's tumor is different, but everybody's white cells are pretty much the same. So there are lots of good antibodies, and you can achieve very, very accurate depletion of white blood cells using these antibodies. But you have to do something to the cells to get rid of them, because you're really getting rid of millions of white blood cells to find one little CTC that's hiding in a corner. The first step of this microfluidic chip is to get rid of the stuff that's small. So unconjugated beads, antibodies, platelets, red blood cells are just removed through kind of a filtration. This is what we call a Pachenko or hydrodynamic cell sorting. All the little guys come out here, and out here come only the circulating tumor cells and white blood cells. The second part of the chip is actually the brilliant innovation here from Mehmet's group, which is called inertial focusing. It turns out if you put a big magnet on cells and they bump into each other, they never really get to the other side. If you have a flow which has these little wiggly circles, there are inertial forces from the walls that press cells towards the center. And what you end up with after this is you end up cells in a single row. So you start here with a big mass of cells going through this channel in laminar flow. By the end of these wiggly channels, they're all in a single row. And once cells are in a single row, it's relatively easy to apply a magnet, and you separate CTCs from white blood cells. So again, this is the engineering genius here, if you will, is to line these up so that a single magnetic bead is enough to separate a white blood cell, and do this at high efficiency. So this is what it looks like in real time. If you tag the CTCs red and the white blood cells in green, you can see them flow microfluidically through this device. This is what the device looks like. It's now mass produced, and hopefully once commercial will be about $20 per chip. It's a disposable chip, and the blood comes in here, the product comes out here, and it's symmetric so that these two are equal, if you will. You can process twice as much blood through one chip. And this is the hydrodynamic sorting by size. This is the inertial focusing to line up the cells, and then the magnet comes in here. This is how the blood plugs into this chip. You basically have an adapter, and again, you can see the blood flowing in here and coming out here. We now have an automated machine that will process. Blood comes in here, out comes the red cells, out come the white cells, and then the product is in between. And this is now a machine that takes about 1 hour to process 10 mils of blood. You can hit the on switch and go get a cappuccino, and hopefully it comes back. And the rate of processing, again, is 10, or could be as much as 10 mils per hour. And we can process about up to 30 mils of blood. Typically, we process 10 mils of blood through the device. So a couple quick applications. The first one, of course, is can you do this? Can you apply this to look at mutations? And the answer is yes. This is work by Tilak Sundaresam, a postdoc in the lab working together with Zhong Li and John Iafredi. You can pull out cells, and the nice thing about the cells is they're not attached to a platform, they're in solution. So now you can stain them for a cell surface marker of choice. There's a nice green cell that we can pick. You can pick a single cell and then subject it to next generation sequencing. And here you see a heterozygous PIK3CA mutation in the case of breast cancer. So you can identify the cells that way. We're trying to find less painful ways of picking individual cells, but micromanipulation certainly works. And that's something we've done a lot. And you heard from David Miyamoto yesterday doing a fair amount of RNA sequencing in prostate cancer by picking single cells like this. Now, the real interesting thing about circulating tumor cells is the ability to monitor cells in real time as they evolve. This is work that was done by Min Yu when she was in the lab together with a breast cancer oncologist, Aditya Bardia. And this was tackling the question of EMT. EMT, epithelial to mesenchymal transition, is a problem. It doesn't have genetic markers. It's really a cell fate change that's defined by protein or RNA markers. Here we're using a particular RNA in situ technology which has been developed by Panomics, an Affymetrix company, and ACD, another company. So these are branch-chained oligos. And you can actually combine 8 different epithelial markers or 3 or 4 different mesenchymal markers, color code them, and get a quantitative readout of how much epithelial gene expression versus mesenchymal gene expression you have. And you can score these. So this cell is all E, this cell is all M, and these guys are in between. So what happens if you look at a patient over time? This is a very, very informative case of a patient who had metastatic estrogen receptor positive breast cancer. And what happened is that this patient had a PIK3CA mutation in her cancer. She was treated with investigational PI3 kinase inhibitors as part of Keith Flaherty's Tremere Center. And you can see that she responded initially for about 6 months. So the total number of CTCs dropped. But what's more interesting than that is that the characterization of these CTCs changed. So initially, all the cells were mesenchymal. They were blue. And as she responded with this new therapy, the mesenchymal cells disappeared, and what most of the cells that were left behind were epithelial. And as the tumor became resistant again, it acquired more blue. And by the time it was resistant, we're back to mesenchymal cells. At this time, her physicians decided to give her adriamycin chemotherapy. She had a short-term, a dramatic response. We're back to epithelial cells again. And again, as the tumor rebounded after the chemotherapy, they became mesenchymal again. So this gives you a sense of just how powerful the treatments we have are and how they induce changes in the tumor that we're treating, both genetic as well as potentially epigenetic here. So this was the motivation for the first story that I'm going to tell you, which is, aside from looking at a fixed snapshot of these tumors, can we actually study them in a way that's more functional? And this is a product of a work of Min Yu, who's in the lab now, has her own lab at USC. And she was very, very diligent and managed to take these cells and eventually grow cultures of circulating tumor cells from women with hormone-positive breast cancer. You can see it's painful. You can see the number of days that it takes to grow these cells. They grow in low serum and under non-adherent conditions, in hypoxic conditions as well, and they need a lot of loving care. Having said that, you can actually take these cells and inject them into a mouse mammary fat pad and give rise to a tumor. In fact, if you look at these cells in vitro, it's kind of interesting. Some of them maintain estrogen receptor expression in the primary tumor, in the cultured CTCs, and in the tumor generated from the CTCs. Others lose estrogen receptor when they're cultured in vitro, but they regain it when they're reinoculated into the mouse. Now, what you can do once you have a purified population of CTCs like that is you can go wild with the genetics. And every time that we study minute amounts of tumors, the purity is not good, you have small amounts of material. Now we have basically a cell line. So this is a platform that John Iafredi and Zhong Li had established for sequencing 1,000 cancer genes in the tumor cell line versus the primary tumor. And you see lots of mutations. The interesting thing is that many of them are different from the ones that were genotyped at the time that the tumor was first diagnosed. So you may know that at MGH we genotype pretty much every tumor that is presented through our clinics. So we have a background of mutations from the tumor itself when patients first present. And in this case, we went back and checked those biopsies to see how many were new. And what you can see in different patients, we have p53 mutations, PIK3CA mutations, FGFR2 mutations, estrogen receptor mutations. This was actually right at the time that these mutations hadn't been reported yet, so we were very fascinated by that. KRAS mutations. All of these mutations were not present in the biopsy sample. So all of these were acquired during the multiple courses of chemotherapy and targeted therapy that the patient received. How do we know that they're not in vitro artifacts? What we can do in patients like this is we can repeatedly sample the blood. So you can see in this patient, for example, while she was responding to treatment, we were unable to develop a cultured cell line that grew. But once the tumor started growing, we were able to culture a cell line. And then we were able to draw a second blood sample a month later and analyze that again. And for all the mutations that I showed you, the two blood samples showed the same mutation. So we know that they really reflect the state of the tumor rather than an in vitro artifact. The other point of this slide is to show you that this is the window we're talking about. So from month 2 to month 5, this is when the clinicians don't know what to treat the patient with, the tumor is getting worse, and in this case, ultimately, the patient passed away. So if we're talking about preclinical diagnostics, we often have a window that could go from 2 to 6 months when we don't know what to do, and this kind of information would be helpful, but it had better happen quickly because, again, the patient is getting quite sick. So this is a study that we did in collaboration with Cyril Bennis in our cancer center, basically testing a whole bunch of drugs. So this comes out of our cell line screen, but we're applying now single drugs as well as combination drugs, targeting different pathways across different cell lines with different mutations. And you'll notice that the mutations, I marked the important ones here, but this tumor has a PIK3CA, FGF receptor, p53 mutation. That's a mutational context. And it's different from this guy, which has another set of mutations. Again, PIK3CA, but this time an estrogen receptor mutation. Each one is different and has a different concept of mutations, and you see different levels of sensitivity to different drugs and drug combinations. So I'll focus on two that were particularly informative. First are these kind of frustrating estrogen receptor mutations. So these mutations are acquired. You never see them, or almost never see them at diagnosis. But the longer you treat patients with aromatase inhibitors, the more you acquire mutations that activate the estrogen receptor in the absence of ligand. And what that does then, you become independent of all hormonal manipulations, and that's not a good thing that happens. As you can see in the cell line here with an acquired estrogen receptor mutation, we can try fulvestrant, we can try raloxifene. We don't see any effect in terms of killing these cells. We actually see something if we add an HSP inhibitor. And if we add the two together, you can see red here, blue here. This is a sign of synergy or cooperation. The combination of an HSP90 inhibitor together with anti-estrogens actually is more effective in killing these cells. And we can show in the cell lines that it destabilizes the mutant estrogen receptor. What was probably even more interesting was another patient in which 50% of the alleles had a PI3 kinase mutation, and 50% had a fibroblast growth factor receptor mutation. And we're assuming that 50% of the alleles means that these are heterozygous mutations, and they're all present in the same cell, but we obviously don't know for sure. Here what you're looking at is a combination where we're treating with an FGF receptor inhibitor and a PI3 kinase inhibitor, and we see very significant synergy when we add the two drugs together, whereas each drug by itself is not quite as effective. And interestingly, we see a similar level of synergy with the IGF receptor. There's no IGF receptor mutation in this cancer cell line, but the IGF pathway is a very important feedback loop, and if you block that, you get the synergistic effect, and here you can see this effect. And you can see this again if you inject the same tumor-derived ctc line into a mouse, generate a mouse tumor, you can see the tumor grows. If you add one drug, if you add another drug, and if you add the two drugs together, you get a synergistic effect. So at least as a proof of concept, it's beautiful, but what can you do with this? What we would argue is that the heterogeneity of cancer cells increases dramatically with progression, and even though we're of any institution in the world, we rebiopsy patients as often as we possibly can here. You can only rebiopsy so many times, and every time you biopsy one site. So the idea of being able to do this at the level of blood-based analysis is very powerful. And clearly, cancer genotypes evolve during therapies. And it's particularly noteworthy that although we could never test it in the patients in these cases, the tumors that were acquired as a result of selection from drug therapy actually drove sensitivity of these cancers. So these women were never treated with HSP inhibitors together with hormonal therapies. They were never treated with a PI3 kinase inhibitor together with an FGF receptor inhibitor, because the physicians who treated for them never knew they had acquired these mutations. So again, that's kind of an interesting, important lesson for us about how powerful these acquired mutations can be. And again, that they all acquire within a genetic context. No mutation is alone. So you have 3 or 4 driving mutations, and even though we know what mutation is driven by what response may be driven by what mutation, the additional mutations in that tumor may direct the response as well. So this idea of preclinical testing is really appealing. But of course, there's some really important challenges. Getting these cell lines to grow more efficiently. Right now, we're still averaging about 10 to 15% of cases we can draw these lines, and it still takes about 3 months to grow them. So we're still looking for ways to make this a little faster and a little easier. Let me turn now to the second story that I was going to share with you, which is more of a biological story. And that is not so much using CTCs as a marker of tumors and to drive therapeutics, but also trying to understand the world of metastasis. And if you look back at that, there are trends. And depending on which decade you live in, there are different dominant mechanisms to understand metastasis. I would say the dominant mechanism now is the assumption and the understanding based very much on mouse models that EMT is important. So epithelial cells just sit there in a cancer, they undergo EMT, become more fibroblast-like, and then they invaginate or extravasate into the blood vessel, travel around, go to the tumor, and then revert to a more epithelial fate. And this acquisition of EMT is key to having cells move around. If you look back at the older literature and you look back at some pathology literature, it's hard to define EMT because there are fibroblasts everywhere, so you never know if your mesenchymal cell is a tumor cell or a fibroblast. I've shown you some RNA staining of CTC, so that's helpful, and there's some lineage tracing studies that are very helpful as well. But pathologists don't like EMT because it looks like fibroblasts to them. What they do notice is they notice tumor microemboli. So when you look at a tumor biopsy or metastatic tumor, you see clumps of tumors with fibrin and stuff, and it's blocking a blood vessel. And the other model is that basically clumps of tumors just break off, or tumor cells proliferate in the blood, and then basically land in a tumor and start proliferating. These two models are not necessarily contradictory, but they're very different in terms of mechanism. So I'll talk to you about work that was recently published by Nicola Assetto, who now has his own lab at Basel. And the idea is that we found when you start looking at CTCs, you see clusters of cells. So this is a clump of tumor cell. This is a red blood cell. It's attached to the cluster. In case you're a tumor immunologist, I'll tell you right away it's a macrophage. So is this one. But these tumor cells are in various states. Some look pretty good, some are pre-apoptotic, but they travel as 12 cells together in the blood. If we look at these clusters, we see anything from 2 cells stuck together to, in one case, where we saw over 100 cells stuck together. And you wonder, how do these things get around the blood? So it's a big challenge to figure out how relevant are these clusters of cells versus single cells. So what Nicola did was to start modeling this in the mouse. And the first thing he did is he took a mouse and injected breast cancer cell lines into the left or the right mammary fat pad, and he tagged them. So these guys are green, these guys are red. And then he looked, what do you see in the blood and what do you see in the lungs? So in the blood, what you see are single CTCs that are green or they're red, or clusters that are either green or red. So what that tells us is the cells in the blood come from this tumor or they come from that tumor, but they don't just aggregate in the blood, because in that case we would have seen green and white cells together. If you look at the lung metastases from these mice, they're either green or they're red, but they're not both. So again, it suggests that each tumor gives rise to its own lung mets. We did find there was an important paper from Larry Norton and Joanne Massaguet about the idea that tumor cells could travel from one tumor to the other. And with this tagging experiment, we could find that as well, but it was less than 5% of tumor cells. So more than 95% of the tumor cells that we find come from one tumor or the other tumor. So the next experiment that Nicola did was to mix green and red cells within a single tumor. Now you have a chimeric tumor, red and green. What do you see in the blood? And what you see is that single CTCs are green or they're red, but the vast majority of clusters in the blood are red and green. So what that's telling us basically, it's not one cell that comes into the blood, proliferates, and its children create a CTC cluster, that you're really breaking up chunks of the tumor. They're oligoclonal from that tumor, if you will, and they're green and red. And if you look at the metastatic tumors, what you can see is that from both of these single cells and CTC clumps, you see that the cells in the lung, the tumors, are mixed. You see some that are single color and some that are mixed color. And then comes the math. If you do the math, it turns out that 2 to 3% of cells in the blood are clusters. But they give rise, because of the multicolor, to over 50% of the lung metastases. If you look at single CTCs, 97% of the CTCs in the blood are single cells. But again, they only give rise to 50% of metastases. So if you do the math, clusters are rare, but their metastatic propensity is about 50 times higher than single cells. And I won't show you the data, but we actually modeled this, and what you can see is as a single cell lands in the lung, you see massive apoptosis. The cells just don't like it in the lung. They die. And there's a period of time, and then slowly the lung tumors start to grow. If you inject clusters of cells into the blood, they just sit there in the lung. They don't have nearly as much apoptosis. It's almost completely suppressed, and then they start to grow. So at least our model would be that the big difference is that when a cluster has enough survival signals from its friends, that when it lands in the lung, it doesn't undergo massive apoptosis, in contrast to single cells. And I would say that Nicola was able to study this both in prostate and in breast cancer. And if you look at patients who have clusters in the blood, especially lots of clusters in the blood, do much, much worse than patients who don't have clusters or have very rare clusters in the blood. Now, turning to the molecular analysis, we did the same kind of analysis that David Miyamoto showed you yesterday in terms of single-cell analytics. And here what we're looking at is taking blood, 265 single cells or clusters from 79 patients with metastatic breast cancer. We sort them through the chip. They're in solution. We stain in red if you're a white cell or in green if you're a tumor cell. This is what we looked at. We micromanipulate them, and then we do RNA sequencing on these single cells versus clusters from the same patient. So the first lesson here is when you group everything together, single CTCs and clustered CTCs from the same patient look almost identical. There are very, very few differences. They cluster completely by patient. That's good because we're not looking at random sequence in this case. There are a relatively small number of genes that are really different between clustered CTCs or single CTCs from the same patient. And among those, one dramatic one that we decided to study because it really kind of tickled our imagination was Plaque Oglobin. And I knew nothing about Plaque Oglobin. I suspect most of you here know nothing about Plaque Oglobin, nor should you necessarily know anything about Plaque Oglobin, but it's worth a couple slides. So Plaque Oglobin is this unappreciated kind of lonely little protein here, which is part of the desmosomes or part of cell junctions. So it's part of a number of the mechanisms. It's intracellular, but it's part of the mechanisms that tether cells together. So in fact, it's kind of glue. If you look at the literature for Plaque Oglobin, half the papers describe it as an oncogene, half the papers describe it as a tumor suppressor gene. We would argue that it's just glue. It doesn't do anything bad. It doesn't do anything good. It helps cells stick together. So what happens if you take cell lines that express Plaque Oglobin and then you knock down Plaque Oglobin with shRNA or siRNAs? Absolutely nothing to the primary tumor. The primary tumor just grows just fine. What happens if you make a primary tumor in which Plaque Oglobin is not expressed and you look at single CTCs? Absolutely nothing. They don't care. What happens if you look at CTC clusters? They go away. So a primary tumor that doesn't express Plaque Oglobin does not make CTC clusters. And if you then look at the number of metastases, it's way down as well. So it's an interesting mechanism, at least in this particular model. What it suggests is that if you take away the glue, you make it harder for clusters to form. And if you have fewer clusters, the tumor is less metastatic. Again, lots to show. This is based on one cell line. We've reproduced this in a second cell line. The interesting thing as well is if you take normal MCF10A or normal breast epithelial cells, they have lots of mechanisms that keep them together. If you suppress plaque oglobin, they still stick together. If you take 8 out of 10 breast cancer cells and you knock down plaque oglobin, they come apart from each other. So it appears that for reasons that we don't know, a lot of breast cancer cell lines seem to rely on plaque oglobin as a rate-limiting glue, if you will. So to summarize now, what I would say is that what we've learned from these models is that ctc clusters break off as clumps from a primary tumor. They're rare, but they're nasty. And again, if you look at the relative importance of these single cells versus CTCs, we would argue that clusters are rare, but they're not a good thing to have in your blood. Plaque oglobin itself helps to tether CTCs within these clusters and enhances metastatic spread. It's an interesting concept if you go beyond that, because whenever we talk about drugs that could suppress metastasis, it's hard to find them. This is an example of, first of all, a drug target that's not an oncogene. It's an innocent bystander, but if we had a way of suppressing it or suppressing desmosomes or cell junctions, you could argue that you'd have a big effect on metastasis. So you might have a big effect on metastasis, and you would never be able to know that by looking at the primary tumor or the metastasis, because these guys don't care. If you look at the metastasis or the primary tumor, you see areas that are very rich in plaque oglobin and areas which are not. So it's kind of heterogeneous within the tumors. But again, it's an interesting drug target, if you will. So I want to leave you with kind of the last conundrum here, which is that just like Matt told you everything that you forgot in biochemistry, something that I forgot in biochemistry, when I took physiology in medical school, they told us about capillaries. So capillaries are these little guys here, and theoretically cells have to squeeze through them, release their oxygen, and then move on. So like, what are these clusters doing? You know, imagine a cluster. A cluster of tumor cells breaks off from a tumor, let's say, in a liver met. It has to travel through the right side of the heart, go through the lung capillaries, back to the left side of the heart, go through the capillaries in your fingers, and then back to the antecubital vein from which we draw the blood. So these 20 cell clusters get around. You know, how does that happen? So either it doesn't happen, but I've kind of showed you that it really does, and they break up from tumors, they don't aggregate in the blood, they really get around. Or we have some channels here, which are less small than we think. Now, you could imagine metastases might themselves have vasculature that's larger. You can have some bypass tracts. But physiologically thinking, we don't like to think about this. So I'll leave you with a picture of a little movie. This is what you get for working with engineers, particularly Fatih and Mehmet. And this is an experiment in which what you're going to see is a little cell that just went through this channel. This is the diameter of a CTC, so a diameter of a capillary. And the pressure is exactly the same as you would calculate across a capillary bed. You can see this big guy coming through here. It squeezes in single row and then reforms at the other end. And you can watch this over and over again. It's actually kind of amazing. But there is amazing plasticity in how these things are kept together and how quickly they go through. So you would imagine that some clusters get stuck in capillaries and then they start growing. Other clusters seem to be pretty happy as they are, and they get through in a way that you never would have guessed under physiological conditions. Again, something that kind of breaks some molds in terms of our assumptions of how things get around in the vasculature. So let me end by saying this is really team science, or the new word now is convergent science. Convergent science is defined when half the investigators don't speak the language as the other half. So we have engineers. Mehmet Toner runs our engineering team together with Shannon Stott, another faculty member, and Ravi Kapoor. And again, it's been a tremendous pleasure working with engineers who basically make whatever devices they want to make, and you don't have to buy a kit or you don't have to kind of make it up yourself. It's been a phenomenal partnership. Our molecular team is run by Shamala Maheswaran, now David Ting, who's on the faculty as well. And the work that I showed in prostate cancer was David Miyamoto, who spoke yesterday, and Eric Zhang. The breast cancer work was done by Min Yu and by Nicola Aceto, who now has his own lab. Clinically, we've worked with Aditya Bardia in breast and Rick Lee in prostate. And again, we've had some great collaborations from Sridhar Ramaswamy's lab and Toshi Shioda for sequencing computational work, and with our pathology colleagues. And this has been supported by a number of sources, both federal, as Stand Up to Cancer, as well as by Janssen. Thank you very much.
Do the presence of CTC clusters correlate with anything you can measure in the blood, let's say immunoglobulin levels, albumin levels, thrombocytosis? Did you check, or is there any correlation? I'm just curious whether there is anything else you can interfere with in order to prevent the formation of these or existence of the clusters.
So the question is, what else correlates with these CTC clusters? We haven't looked at proteins in the blood. And again, the numbers are relatively small, and most of these we haven't had access to for some time. The only thing we could see correlation-wise is that patients who have a lot of CTC clusters often have fairly advanced disease. So it's really kind of a tumor load, tumor aggressiveness type of status. But even when we correlate for the number of metastatic lesions, we see a dramatic correlation with outcome in terms of the more clusters you have, the worse you do. I showed you the curve for breast cancer. In prostate cancer, it's even more scary, where again, 1 or 2 clusters in the blood really correlates with shortened survival.
And did you check their sensitivity, you might have mentioned, of the clusters versus individual cells in your drug screening?
So most of the work that I showed you on clusters is just imaging. When we culture cells, I would say that we've cultured cells from a single cell. We've cultured cells from patients who have lots of cells. Generally, if a patient has clusters, it's a better sign that the culture will take. But once the culture takes, we can't tell whether it's single or not. Okay. And the CTC lines tend to grow as clusters, in fact, because they're kind of, again, in 3-dimensional space. But we can't impugn anything about the two.
Okay, I'm wondering if there's any way to try to explore the relevance of clusters and plaque oglobin specifically in seeded micromets. In other words, the work you're showing and all the images kind of implied this is all in circulation, but the success of metastasis, or success or not, lack of success, may have everything to do with how that sort of seeded micromet that could be dormant for some time thereafter behaves. Is it possible to isolate clusters, infuse them in tail vein, wait a couple weeks, let them lodge or not lodge, then try to understand the relevance of downregulation of plaque oglobin or other targeting strategies?
We haven't done that. And again, the individual tumors are very heterogeneous in terms of high plaque oglobin, low plaque oglobin regions of a tumor. We hypothesize that when a tumor block breaks open, it's held together by this plaque oglobin-rich area, and these may be the things that kind of fall out into the circulation. The farthest we've gone in modeling is introducing cells into the tail vein of a mouse with or without plaque oglobin. And again, the differences there are very, very dramatic, but that's because you knock down plaque oglobin and the clusters go away. Yeah, right. We haven't been able to do this in real time once the tumor is initiated.
Yeah, right.
Plaque oglobin itself is intracellular, so it's not really amenable to therapy other than shRNA-mediated knockdowns. And it's also a relatively slow turnover protein once it's made. But I think it points to desmosome cell-cell junctions as something that could be targetable through antibodies, and hopefully that's where the field would go. And again, the idea is that in a normal cell, you might disrupt cell-cell interactions through multiple mechanisms. Cancer cells may be more susceptible to desmosomes.
Yeah, I think it was a great presentation. And this is following up what Keith was mentioning. I'm sure that you have thought about doing an inducible knockout mice and crossing with your green and red cells and see whether you can prevent this in those situations? Or have you thought about doing something in vivo to demonstrate that the lack of plaque oglobin can prevent this induction of metastasis? And the second thing, if you know the mechanism by which, when you have the CTC cluster, you increase so many times the plaque oglobin expression.
So to your second question, our assumption, I mean, we don't know, but our assumption is that expression of plaque oglobin dictates the cells that stick together and form a cluster, rather than clusters inducing plaque oglobin secondarily. So our guess is that, again, we don't know, but we're assuming that there's so much heterogeneity in plaque oglobin expression that if you look across a tumor, you'll see 10 cells with incredibly high expression, and everybody around it doesn't express it. So it's tempting to think that that's your cluster in situ, and then when it breaks off, that's how it happens. But we don't have proof for that. In terms of knocking down plaque oglobin, we did this using shRNAs because the fact that if you knock down plaque oglobin with shRNAs, it doesn't prevent or doesn't affect the growth of the primary tumor, meant that we could then look at metastasis. Had it affected the growth of the primary tumor, we would have to do an inducible construct, as you mentioned.
Well, we're thinking more in mice that has inducible knockouts. Have you thought doing, or I don't know if it's available?
You mean congenitally?
Yes.
That we have not done, no.
Okay, thank you.
Very interesting talk. Have you used your dynamic monitoring to look at immune profiling? Or you probably have?
Well, we're trying. With great excitement, we noticed that a lot of our big CTC clusters often have white blood cells attached to them. And it's such an irony because initially we were so annoyed. It was like, you think they're pure and what are these damn white cells doing there? And then we talked to a couple friendly immunologists and they told us, forget the tumor cells, it's all about the white cells that are accompanying it. So in a handful of cases, we're able to take these, do RNA sequencing, and that's where we got the signature of tissue-based macrophages for the dominant signature with these CTC clusters. But we don't have enough, and it's a question of numbers now that we have to accumulate enough of these clusters with attached white blood cells to really be able to have some kind of real answer.
What about even simple PD-L1 monitoring on the tumor cells?
We're working on that. The problem with PD-L1 is that it's expressed, of course, in white blood cells in the circulation. So we're now working on trying to get good PD-L1 staining that is specific to a tumor marker. So again, there's a fair amount of imaging that you can do in CTCs where you can do, whether it's spectral or other complicated types of imaging, define the cells and ask within those cells how much PD-L1 is expressed. And so far we're still working on the conditions that would be pure enough to allow us to do that.
Hi, have you noticed a difference in the profile of the circulating tumor cells once they're taken from the circulation and then cultured? Is there any change in the profile or what's expressed?
We've been encouraged by how similar they are. So what we've done, again, now you're getting into technology differences. So what we do is when we pick a single cell, we have a particular protocol for single cell RNA sequencing. So you start with minute amounts of template, and then there are a number of fairly complex protocols to get a whole transcriptome worth from a single cell. We then take our cell lines, dilute them down to that same level, and then compare the two, and they're virtually identical. So that's been reassuring in terms of the reliability of these. And again, those cultured lines look weird. They don't look like MCF7 cells, so that comforts us to think that they really have kept some of their weird characteristics.
Dan, forgive me for going back historically. The great French morphologist, hematologist Marcel Bassis, who did such magnificent microcinematography many years ago, showed the unit of erythropoiesis being a central, almost dendritic, but central macrophage surrounded by a corona of erythroblasts, and actually looked at ferritin and so forth in electron microscopy. And it was at that time, as a Callow postdoc, that shows you how many years ago it was, that he also happened in a number of experimental circumstances to view tumor clusters that were actually circulating in a rat model, and found that macrophages and monocytes were therefore also attached. It'd be interesting to go back and review some of that early microscopy, and that's all available at the Institut Pathologie Cellulaire in Paris. And it's remarkable how you've taken us, of course, another quantum, but how some of those early observations that have been published might be interesting as a further illumination of what may in fact be the extant model as you're so beautifully demonstrating it.
It's a beautiful historical view. It also has a funny side to it. The first CTC described was by someone called Ashworth in Australia, who did a camera lucida drawing of a CTC, which looks very much like what we see today. The funny side is that we get two kinds of reviewers for all our manuscripts: those who think that it's cool and very interesting, and those who say, well, this was discovered 200 years ago and there's nothing new here. So hopefully you belong to the first category.
Okay, last question, Jeff. So to try to kind of bring yesterday and today together, Sridhar, who you collaborate with, has been studying these dormant cells, as you know. And some of the characteristics of those dormant cells reminded me of these cell clusters that you're looking at. And specifically, you know, their slow growth, the specific conditions in which he has to apply in vitro, and the fact that sometimes you could see these clusters. So I'm curious about whether you have any data to indicate whether these cells that you're seeing as these CTCs actually represent the dormant tumor cells that actually present huge problems for therapy, and they break off. They go into the blood. Now they land in a new location whereupon they can be reactivated as a metastasis. Do you have any evidence for dormancy in these clusters?
So the two parts to your question. In terms of looking at the markers that Sridhar has defined in terms of, for example, the notch activation,
Right.
in particular epigenetic marks, those we've looked
It was
at.
actually AKT, I think, was he was saying is the main one. Yeah.
Yeah, that's harder to look at it in vivo, but looking at those in our cell lines as well as in primary cells, we don't see those same particular markers. Having said that, the whole field is really converging because clearly what you see is these cells do give rise to local cells in distant organs where they're called disseminated tumor cells, and they sit there for long periods of time, whether it's a mouse or a human. So clearly, whatever mechanisms allows them to travel in the blood a couple times around, because the half-life is in the 10 to 20 minute range in the blood, they circulate for a while before they land, and then they have to survive that process. Whatever enables them to do that obviously has to transition to a more proliferative phenotype.
And have you ever looked at SOX10 by any chance?
We've looked at SOX10, which is expressed as one of the genes that we find enriched in CTCs compared to primary tumors, yes.
Yep.
Thanks very much.
Thank you.